Hello everyone, I am Dr. Raman. In our last class, we had discussed topics linkage, crossing over, gene and chromosomal mapping, and different problems, numerical problems related to these topics. Okay, today we will just continue with the practicals and we'll start practical number 9, that is gene interaction. Okay. Gene interaction, it is just you can, in very simple words, if you ask me what is interaction, it is mean influencing two genes. Okay, in which way two genes are influencing each other or other genes, that is the gene interaction. Okay, the gene interaction, it is of different types. Okay. The major classification, it is mainly based on the epistasis. One kind of gene interaction, it is non-epistatic gene interaction, and other is epistatic gene interaction. Okay. Uh, if we start with the non-epistatic gene interaction, means that in this particular kind of gene interaction, there is no epistasis. Okay. We'll start with the gene interaction. Okay. The expression of an allele will alter the expression of one or more known allelic genes. The classical example of non-epistatic gene interaction is the comb shape. Okay, this is very common example, the comb shape in the case of poultry. Okay, comb shape. This is the example of non-epistatic gene interaction. Okay, where gene R is the rose while gene P is P, and genes R and P, when they are together, they interact to form a walnut. Okay, these are different kinds of, you can say, the comb shapes. Okay, comb, it is just, for example, this is the face of a bird. Okay, and above that they have different kind of combs. Okay, these are combs. Okay, so these combs, they just seen because of these particular genes and their interactions. Okay, we have R. R gene is for the rose comb. P is for the P comb. R plus P. When they both are present, then that form walnut. Okay, these are the three very basic forms, and in the condition where R and P both are absent, and both are absent, that particular situation is single comb. Okay, so if these are not present, then that is a single comb. Okay, yeah, we'll start again. R is rose comb, P is for the P comb. When gene P and R, they are together, they interact to produce the walnut comb. In classical ratio, um, in F2. Okay, when we just cross these, and we will get 9331 ratio. Okay, this is the classical example and the classical ratio for non-epistatic kind of gene interaction. Okay, we'll move with the example Example in fowl, example in the case of fowl, two pair of allele, that is PR and PP, they regulate the shape of the comb. Okay. In the cock, the rose comb is mated with the Brahma hen with peacock. Okay. Uh, Always just keep this in mind. If at a place, if it is written like, uh, see, if it is written like a rose comb, then that shows it is a homozygous. Okay. If it is not homozygous, then examiner or the person will mention that it is a heterozygous condition. Okay. So if a particular individual, that male, is having rose comb, then that means he is having the capital R's. Okay. This capital R1 male is mated with the Brahma hen, which is having PCOM. PCOM means capital P's. Okay. So if this particular individual, this male and this is the female, if male is having rose comb, then that means he must be having this recessive gene of PCOM. Okay. Similarly, if he is having PCOM, he must be having this recessive R gene for the rose comb. Okay. If these are the dominant genes, are capital R and capital P, then only this particular individual is showing the PCOM. This is the rose comb. Okay. So if we are mating them, then we will get the individual which will be having R, capital R, small r, capital P, and small p. We'll get this genotype. Okay. So this is our F1. Okay. So if we cross this with the similar genotype, this means either it is selfing, or it is interbreeding. Okay. If in any question or in an example, the person is saying that these birds or these animals are intermating, and that shows that we are crossing the similar genotypes with each other. Okay. So if we cross these, we will get this 9 is to 3 is to 3 is to 1 ratio, where nines are the walnuts, threes are the rose combs, threes are the pecans, and one is the single comb. Okay. So we'll move forward. We'll move forward in our example. The chick produced had a walnut comb. Okay. Uh, After crossing capital R, small p, if we are crossing it with small r with capital P, we will get the individual which will be having capital R, small r, capital P, small p. Okay, so he is having capital R and capital P. So both of genes, if they are together, they'll form the walnut. Okay, so the chick produced had a walnut comb. So this is the chick which is produced after crossing these two, rose comb and pea comb individual. Okay, so this is the walnut comb. So after later on inbreeding, it produced 19 walnut, 6 pea comb, 5 rose comb, and 2 single comb. Okay, 19, 6, 5, 2. You will say that, sir, you had told that the ratio is 9331. Okay, so it is just the double, approximately double of the ratio. Okay, just 1 plus 1 minus. Okay, so this is just the double of the that particular ratio. If we divide this by 2, this by 2, 5 by 2, this by 2, and then that will give us approximately 9, 3, 3, 1. Okay, we move further. The F1 is double heterozygous as uh, in F2 9331 ratio is obtained. Okay, F1 is double heterozygous. We know that it is having capital R, small r, capital P, small p. So that particular individual is having uh, heterozygous condition on both the genomes. Okay, both the alleles. Walnut has one dominant gene, and at each locus, yes, we know one capital and one capital R, one capital P. Okay, he is having one dominant gene at both the locus. Okay, this is the kind of individual which will be having walnut com. Okay, while rose and pea com will have a one dominant gene. Okay, this is R. If he is having one capital R, that means he is having the rose com. Okay, we'll have 3 individuals of this kind of genotype. Okay, pcom, the one with the pcom is capital smaller capital P, then dash. Okay, that capital here it is small p, then you just change it to the capital P. Then single com, it is with small p. Every gene is in the recessive condition. Okay, so if someone asks what is the genotype of the cock, that is capital R, small p, that is for the cock. If for the hen it is smaller, capital P. That I had already told you, that if the rose comb is written, then you must always go with the homozygous condition. If it is heterozygous, then in any way examiner or person will mention that. Okay, but if that is not mentioned, you just have to be cautious. Okay, just open your mind. If you're doing some kind of numerical using this and it's a multiple choice question, the answer which you're getting, it is not into your uh, calculated ones, then you must go with the other heterozygous condition. Okay. Next is epistasis. Epistasis is when a gene at a locus masks the action of gene present on another locus. This is called epistasis. The gene whose effect is masked, it is the hypostatic, other is the epistatic gene. Epistatic will mask the effect of the hypostatic gene. Okay. In F2, the number of the phenotypic classes for are the 4 or less than 4. Okay. In the case of non-epistatic kind of interaction, we had seen that the ratio is 9:3:3:1. That is, we will get the 4 classes. But in the case of epistatic interactions, these 4 classes will merge in any way, then we will get the 3 or 2 classes. Okay. First is dominant epistasis. Dominant means dominant allele at one locus A suppress the expression of other locus regardless of its allelic condition. Recessive epistasis, one recessive homozygous suppress the expression of another locus. Duplicate gene with cumulative effect means dominant condition is either locus produce the same phenotypic effect. Duplicate dominant gene. These all interactions you had read in your theory. I'm not going into much detail. Okay. Uh, these are the different values. If we check the classical ratio, it is 9 is to 3 is to 3 is to 1. If we go with the dominant epistasis, it is 12 is to 3 is to 1. In the case of recessive epistasis, 9 is to 3 is to 4. Okay, now we are not getting all the four values. Okay, and now we are having 3. This is also 3. Duplicate gene with cumulative effect, that is, you always just try to remember these ratios. um, we can ask these in MCQs, that what is the ratio for dominant epistasis, what is the ratio for the recessive epistasis, or what is the ratio for the duplicate dominant gene, recessive gene. Okay, so you just always keep these values in your mind. Duplicate dominant gene is 15 is to 1. Duplicate gene with cumulative effect, that is 9 is to 6 is to 1. Duplicate recessive gene, that is 9 is to 7. Okay, now 3 is to 3 is to 1. These both merged. In the case of dominant recessive interaction, we'll get only two classes, that is 13 is to 3. Okay. The example is the feather color in the fowl depends on the action of two loci, prevents the expression of the gene color of another independently assorted locus and produce white color. When the recessive condition exhibit at the inhibitor locus, the allele of hypostatic locus expressed IIB produced colored, and IIBB produce white. Okay, if at any locus, if this particular individual is having capital B, okay, then only that particular individual will show the color. Okay, otherwise at every locus that individual will show the white color. Okay, let me show you. If this is IBIB, okay, IBIB, these are the white individuals. If we are crossing these whites, we will get 9 is to 3 is to 3 is to 1 ratio, where nines are of this genotype, they are white. This is also white. If only B is there, then that is colored. Otherwise, capital B is there, then it is colored. Otherwise, it is white. Okay, so in this condition, or in this particular case, we'll have the ratio of 13 is to 3. And this kind of interaction, 13 is to 3, is a dominant recessive interaction. Okay, so if someone asks feather color of the fowl, or white or colored condition in the case of feather color of fowl, uh, it exhibit which kind of gene interaction, that is the dominant recessive interaction. Okay, because it will just give 3 is to 13 is to 3, uh, which are this is the phenotypic ratio. Okay, not genotypic. Genotypic ratio, they are still 9 is to 3 is to 3 is to 1. You can say that we have 4 major classes genotypically, but phenotypically they are having 13 is to 3. Okay, the chance of white progeny is 13. Okay, at 13 places we have white, and only at 3 places, 3 out of 16, we are having the uh, colored. Okay, number of white homozygotes. Okay, white homozygotes are, okay, one will be here, one will be here, and one will be here. So total will have 3 white homozygotes. Other rest will be the heterozygotes. Probability of white homozygote in F2, white homozygote, these are number of homozygotes are 3, so the probability is 3 by 16. Probability of white homozygous among white. Okay. They are 1 by 3 into 13 by 6. That is 3 by 30. Okay. And only homozygous at both loci. Okay. That is 1 by 3, 1 by 3, 1 by 3. That is equal to 3 by 3. Okay. 3 by 13. Sorry. This problem, it is already solved in the case of example here. Okay, no need to write that again. Okay, we had already covered that. Okay, next is our 10th exercise, that is estimation of gene and genotypic frequencies in Hardy-Weinberg law. Okay, from this portion we'll start our population genetics. Okay, above that we were having our genetics portion. Now this, from this exercise 10th, this is your population genetics. Okay, the difference between population genetics and Mendelian genetics, you always know that population genetics, it is mainly based on the population. Mendelian genetics, it is for the individuals. Okay, that is not for the whole population. Okay, and rest differences you all know. Okay, in population genetics, it is the study of genetics at population level. Mendelian population, it is a group of sexually reproducing organism. They are relatively closer degree of genetic relationship. Okay, residing with the defined geographic boundaries, uh, wherein inbreeding occurs. Mm, and that is known as the Mendelian population. Okay, we are having inbreeding, we have the close related genetic relationship. Okay, we have a well-defined geographic boundaries. Okay, so they and yes, all are sexually producing organisms. Okay, so these are the different characters of the Mendelian population. Gene frequency. Okay, now we have two major terms. One is gene frequency, other is the genotypic frequency. Okay, gene frequency means what is the frequency of a gene. Okay, in our last lecture, or in the last practical, uh, we had studied the ROSCOM, PCOM, and all. Okay, so if I have to check how many percentage of population is having capital R, okay, that means I am just taking only this R into the consideration. So R is the gene or allele. Okay, or I have to check the concentration or the quantity of P gene into the whole population. So that is the gene frequency. Okay, but if I am talking about the rows count, okay, the rows count, for rows count, one individual has to have a R, other blank. Okay, so now this is the genotype. Okay, so this will give us the genotypic frequency. Relatively rarity and abundance of a gene in comparison to its alleles at the particular loci or locus, that is the gene frequency value, ranges from 0 to 1. Okay, the relative portion of favored genotype in a comparison to other genotype present, that is the genotypic frequency. Okay, this color is a phenotypic character. Yes, this color is a phenotypic character. You just don't confuse with that. But if I am talking about this particular sequence, okay, that is the genotype. Okay, so that will give us the genotypic frequencies. Next is the Hardy-Weinberg law. If the gene and genotypic frequencies will remain constant from generation to generation, in an infinitely large inbreeding population, which is mating at random, there is no selection, migration, mutation. This is called Hardy-Weinberg law. Okay. Hardy-Weinberg population or the law, you always have to remember, okay, that this whole line, you just always have to remember. Okay. Not, I don't want that you just cram it. Okay. It has some basic points. Okay. I'll give you the basic points. So try to remember uh, those points. This whole definition will always be in your mind. Okay. Hardy-Weinberg law. Okay. If both gene and genotypic frequencies will remain constant. Okay. First point is gene frequency, genotypic frequency will remain constant from generation to generation. Okay. Gene frequency, genotypic frequency, they always remain constant from one generation to the next generation. Okay. So in every generation, this gene and genotypic frequencies, they remain constant. The population size, it is large population size, and they are interbreeding. Okay. They are interbreeding. Okay. And the mating is a kind of random. Okay. It is the random kind of mating. Okay. Now again, gene and genotypic frequencies, they remain constant from generation to generation in a largely random mating population or large interbreeding population, which shows random mating. Okay. There is no selection, no selection, migration, and mutation. Okay. There is no selection, migration, and mutation. Okay. Again, gene and genotypic frequencies, they remain constant from generation to generation. Population size is large. It is interbreeding population with the random mating, and there is no selection, migration, and mutation. Okay. This shows the Hardy-Weinberg law. This is the Hardy-Weinberg law. Okay. The Hardy-Weinberg law depends upon the following kinds of the genetic equilibrium for its fulfillment. Okay. If a particular generation or a population, it is just showing these characters, we say that this particular population is into the genetic equilibrium. Okay. That means this particular population is into the genetic equilibrium. Now, what are the characters for a population to be into the genetic equilibrium? The population is indefinitely large, made at random, no selection, okay, no immigration, no emigration, population is closed, no mutation, okay, meiosis is normal, so that the chance of only factors operative in gametogenesis, okay. Testing a locus for equilibrium. Hardy-Weinberg law estimates the relationship between the gene and genotypic frequencies. If P and Q are the frequencies of allele A1 and A2 respectively at the locus, then the genotypic frequencies at the square of gene frequencies um, in the equilibrium population, that is P plus Q square is equal to P square plus Q square plus AQ plus 2PQ, where this is the genotypes for that particular value. Okay, this is very simple. We have two genes, one is P and Q. Okay, if for a particular trait we have 3 4 genes, then we'll just say P Q R S T. Okay, n number of genes. Okay, but if they are into the equilibrium, then they just show this kind of relationship. Okay, where P plus Q is equal to 1. Okay, and P square plus Q square plus 2PQ, that is also equal to 1. Okay, if this is equal to 1, then that particular population is into the Hardy-Weinberg equilibrium. Okay, or genetic equilibrium. Okay, where this is the genotypic frequency for the dominant, this is the genotypic frequency for the recessive, and this 2PQ is the frequency for the heterozygote. Okay, P plus Q is equal to 1, P square plus Q square plus 2PQ is equal to 1. Okay, this is dominant character, this is the recessive gene, this is the heterozygote. Okay, if we are considering it is the dominant, this is the heterozygote, and this is the recessive. Okay, so the n is total number of population, total number, that is D plus R plus H. Okay, that will give us the n is total. Okay, genetic frequencies addition. Okay, so the genotypic frequency are the square of the gene frequencies. Okay, P, if we are just squaring it, that will give us the genotypic frequency. Here it is that it is the binary equation. Okay, expected number of different genotypes. We will be having P square, 2PQ, Q square. Okay, these are the different kind of genotypes. And move further. The method for testing whether the population it is in equilibrium or not. Okay, one is chi-square method. I'll just clean this. One is the chi-square method. Chi-square is O minus E square by E. You had read this in your stat course. Okay, what is chi-square? Where O is the observed and E is the expected. Observed minus expected square divided by expected number of the genes or genotypes, that will give us the chi-square value. Okay, and then using the degree of freedom, we check that this particular test is significant or not. If it is significant, okay, if it is significant, the population is said to be not in equilibrium. If it is not significant, then we say that population it is in equilibrium. Okay, the difference between the genotypes, observed and expected numbers. Okay, in random mating the population, if H, R, and D, this is the second method. The proportion of heterozygote, recessive, homozygote, and the dominant homozygote individual respectively, then H square is equal to 4 D R. Okay, if this is like this, H square is heterozygote, D is the dominant, R is the recessive. If it is like this, then it is the equilibrium population. If this is not there, okay, then it is not in equilibrium. Okay. Single locus with two alleles, no dominance, codominance. Okay. In this particular situation, you can say that generally, if there is no dominance or codominant characters are there, then we just follow this particular formula. Okay, gene for calculation of the gene frequencies. This is not capital, and this is the small p. Small p is the gene, small q. Okay, so to calculate the small p, what is the formula? That is twice of d plus h divided by twice of n. Okay, this is the formula. Or what you can say, we divide these all with 2. Okay, that will give us the d plus h by 2 divided by n. Okay, how we get this formula? We divide these all with 2. Okay, d by 2, h by 2, n by 2. Okay, this 2 will cut with this 2, this 2 will cut with this 2, and h by 2 will come here. Okay, this is h by 2. Okay, in this way you can calculate the value of p, and for the calculation of q you just have to remove this d with r. Okay, because d is p square, r is the q square. Okay, for the recessive character we'll use this value, q square value. Okay, for this dominant we use this p square value, and heterozygotes are 2pq. Okay, in the case of complete dominance, uh, when the dominance is complete, a single locus with the 2 alleles would show only 2 phenotypic groups. Okay, one are the homozygous dominants, heterozygotes are indistinguishable. Okay, we had already seen this kind of characters in our last uh, at the time when we were just doing the gene mapping. Okay, at that time we had seen, okay, complete dominance, incomplete dominance, and no dominance. Okay, so in that particular situation, we'll just only have the homozygous groups. Okay, heterozygotes are not just generally seen. Okay, uh, multiple alleles, they also just show that random traits, that Hardy-Weinberg, they also follow Hardy-Weinberg principle, but to follow that completely, this particular genes, these particular genes have to be into the random mating situation. Okay, some important points: if a relationship that is p plus q square, that is p square plus q square plus 2pq, this holds good, then the population it is in the Hardy-Weinberg equilibrium. If the population it is in Hardy-Weinberg, then gene and genotypic frequencies will remain same for both males and females. Okay, this is important point, that if a population it is into the Hardy-Weinberg equilibrium, then the gene frequency it is same for both gene and genotypic frequency. They are both same for both male and females, generally for the autosomal traits. Okay, if the multiple traits they are not showing, they are not into the random mating situation, then this may not be equal to 1. Okay, the calculation of gene frequency for autosomal trait is same for calculation of gene frequencies for in the sex, for the female sex linked genes. Okay, uh, the differences in the gene frequency between two sexes is reduced by half of the previous generation, but of opposite sign. Okay, the gene frequency that will just become half from one generation to the next generation. Okay, but with the opposite signs. Okay, there will be 3 genotypes in the females, 3 genotypes in the males for the autosomal traits. Okay, this is important. There will be three genotypes in females, three genotypes in the case of males for autosomal traits, whereas two genotypes in the case of females, and two genotypes for the sex-linked traits. Okay, the gene and genotypic frequencies is same in males for the sex-linked traits. These are the important points which you have to remember. Now we'll start our numericals. At what allelic frequency does the homozygous recessive genotype AA become twice as frequent as the homozygous genotype in the Hardy-Weinberg? Okay, but they are saying that this has to be twice the homozygote. Okay, this or twice of this, small a small. Okay, in which condition this is seen that what could be the allelic frequency at that particular situation? Okay, so if it is twice, then we generally see that 1 is to 2 is to 1 ratio. It is only seen when P is equal to 0.5 and Q is equal to 0.5. Okay, you can calculate that. Okay, uh, but you just try to remember that if this is the twice of this, then the gene frequencies they are this. Okay, because in that only that situation that will exhibit 1 is to 2 is to 1 ratio, and only in that form they can just be twice. Okay, the population consists of 25% of homozygote dominants, 50% of the heterozygotes, 25% of the homozygotes. Okay, the thing which I had told you here, this is the you can just say that it is the calculation for that. Okay, what is the gene frequencies? Okay, I had already given you the formula for P is equal to D plus H by 2 by N. Okay, this is for P. Now we have D is dominant is 25 plus H by 2 is 50 by 2, that is 25. Total N is 25 plus 50 plus 25, that is 100. Okay, that will give us 50 by 100. So in that particular situation, the P is 0.5. Okay, if P is 0.5, then Q is 1 minus P from Hardy-Weinberg law, where P plus Q is equal to 1. Okay, so P we know that it is 0.5, 1 minus 0.5, that is equal to 0.5. Okay, so that is 0.5. Okay, you can calculate the value of Q from this Hardy-Weinberg law, or you can calculate by changing here the value of R. ठीक है? Here now it is Q. Q is equal to R plus H by 2 divided by N. R is 25, H by 2 is 25, 50 by 2 is 25, and n value is 100, that will give you 0.5. Okay, that this question is very easy. Okay, and in this situation you can say that heterozygotes are twice as the homozygotes. Okay, so for this and this question, they both are similar kind of questions. Next is question number 3. In MN blood group system in humans, it is governed by the codominant alleles. We have total 208 samples we tested for the presence of MN antigens. They are found 119 with M. That is, they have LM. 76 they have MN. That is LM. 13 groups they have LN. Okay, that is N group. Okay, so in that particular question we have to calculate the gene frequencies of LM and LN. Let's consider it is the P, this is the Q. Okay, similarly the value of P is equal to D plus H by 2 by N. Okay, we have D is 119 plus H by 2 is 76 by 2, that is 38 by N. N is total value is 208. Okay, so if we calculate it, that is approximately equal to 0.755. Okay, that is 0.755. Okay, this is the value of P. If you check the value of Q, then it is 1 minus P, that is equal to 1 minus 0.755, that is equal to 0. Is the cursor? Yeah, 0.244 or 245 to be more precise. Okay, so this is the Q, this is the P value for this particular population. Okay, calculate the gene frequency LM. So LM is 0.755 and LN it is Q, and Q is 0.245. Okay, now in our next step, if the gene frequencies of LM is equal to 0.3, if as we had calculated it is LM is 0.755 and LN is 0.245. Okay, but in this second situation, if they are saying that if the value of LM is 0.3, how many individual in the sample size of 500 will be having that MN blood group? Okay, now we have to calculate the gene frequency. Now we are having P value, that is 0.3. Okay, LM we had considered it as P, this is Q. Now P we are having, Q we can calculate 1 minus P. Okay, that is equal to 0.7. Okay, so From the Hardy-Weinberg law, p square plus q square plus 2pq, that will give us the whole population, where p square they are the dominants, this is q square is the recessive, and 2pq is the heterozygote. Okay, now what we have to calculate, that is the number of individual which are heterozygotes. So we have to calculate the value of 2pq. Okay, so 2 multiplied by p, what is the value of p? That is 0.7, and the value for q, it is equal to 0.3. Okay, so that will give us the overall value, that is 0.42. Okay, so 2pq value is 0.42. Okay, or you can say that from 100% population, 42% is are the individual which are having MN blood group. Okay, out of 100% of population, 42%, or out of 0.42 are the individual which are having MN blood group. Okay, I had multiplied 100 on both sides. 1 multiplied by 100, that's 100. 0.42 multiplied by 100, that is 42%. Okay, that will give us the percentage values. And from 42%, let me calculate in 100% of the population. Okay, now population size is 500. 100% of the population, it is 500. 1% is 500 by 100. Okay, now our value was 42%. Okay, in this way we can calculate the number of individuals which are having MN blood group out of 500. Okay, so that is 500 by 100 multiplied by 42. Okay, that is equal to 42 multiplied by 5, that is 210. Okay, so out of 500, 210 individuals were the individual which were having MN blood group. Okay, now we'll move forward. The gene is codominant with allele red color control the color black colors. Okay, as now here they had written that this red color, it controls the black color, means this red color is the dominant character. Okay, as it is controlling this black recessive character. Okay, the heterozygote is red. Heterozygotes, they are also red. That also confirms that red is the dominant one with varying black spots. So means the dominants are red, the recessives will be black, but the heterozygotes will be red with black spots. Okay, this is clear from these two lines. Okay, the total flock size is 150. 150 is the total flock size, where 40 are black, 30 are red, and remaining 80 are spotted. Okay, what is the frequency of genes for red and black? Okay, similarly, just with the same formula, uh, that is P is equal to D plus H by 2 divided by N. Okay, here D is dominant character, that is 30. 30 plus H by 2 is, it is the heterozygote, that is 40 divided by 150. Okay, that is 70 by 150. Okay, that is 70 by 150. For the Q, similarly, at the place of this T, will write the R. R is recessive, that is 40. 40 plus 40 means 80 by 2. Okay, divided by 150, that is 80 by 150. Okay, that if you solve these, that will give you the value of P and Q. Okay, P is the red gene and Q is the black gene. Okay, the pole condition, it is completely dominant to the horn condition. Okay, our last today's question, the pole condition, it is completely dominant to the horn condition. The pole character is dominant and small p will give you the horned ones. Okay, poled. This is the horned. Okay, in the herd of total 400 cows, we have 180, they are poled. Poled is 180. Okay, now it is the single character, that is horned character. So this particular condition is like this: P, P, okay, P, and last form is this. Okay, these three different phenotypic groups were seen with 1:2:1 ratios. Okay, in generally, if we are talking the monohybrid process, okay, 1:2:1 ratio will have the three classes. Okay, so these two will show the, as it is the completely dominant character, and these two will show the similar phenotypes. That means this capital P, this capital P, they'll make the poled group. Okay, only this group will be having the horned character. Okay, so if we are having 180 cows, they are poled. So these two groups collectively make 180, and this group will make the rest of the animals. So horned animals, we are having 220. Okay, out of 400, 180 are the poled ones and rest are the horned ones. Okay, now we have to calculate the frequency for the horned animals, that is 200, 220 by 400. Okay, that will give us the frequency, that is 0.55. Okay, I had already values with me. Okay, so that's why I'm just writing these values, because they can just save the time. Okay, now this 0.55,